parasitology, techniques of molecular biology, and preliminary idea in the application of animal taxonomy and systematics, stem cells, and regeneration, wildlife, and so on. Every one of the topics I know would be very interesting, especially for the students of science who are learning, who were so far learning through classroom deliberations. They are now using a technology that is also, I find it very, very impressive at the number of students participating in this student development program, because that means that technology, that this everyone is can avail the technology and move forward in their academic sphere. So this, I know this is going to be a very interesting, very knowledgeable, and very much very good endeavor, very good development program. 2 weeks for 2 weeks for the students and the teachers. It will be a very good experience for the students as teachers as well, teaching through a different medium and teaching in a different way. So I hope everyone comes out much richer in knowledge, in awareness, and I wish this process immense success. Thank you.
Thank you very much. One. Extremely sorry for the initial inconvenience, because since I have set it in meeting mode according to the convenience of all the faculties who have told that we need an interaction, but we should do it in webinar mode from tomorrow only, or today just after 15 minutes I am setting a new meeting in webinar mode after this inaugural session, so that all the students who are in YouTube can join, because otherwise there will be less interaction. Okay, so here I would like to request Shumana to say a few words. And ma'am Shumana, all of you, I want to introduce today's resource person. Dr. Shuman Bhusan Chakraborty is also here. He is the resource person from University of Calcutta. Later I will introduce him in detail. And Dr. Shaunak Shahu from NIH. Okay, he is also the resource person for another day. And Shumana is our resource person, overseas resource person, as well as collaborator also. I do request Shumana to tell a few words about this program.
Thank you, Papia. Thank you, Professor Bhattacharya, for the nice words. Hello, everybody. This is Shumana Mandal. I'm talking from on behalf of Clarkson University, though I'm talking from my home because we are still not allowed to go to the campus due to the COVID reasons. We are given permission one by one, and our classes will start, thankfully, on 19th August. So Papia contacted me a few weeks back regarding this program, and she said that let's do something that will help all of us, because we are all sitting at home, but the learning should not stop. And I was very excited because I never had done anything like this in collaboration with my Indian friends and Indian collaborators. And of course, Papia is a childhood friend. I cannot say anything. If she asks me something, I have to say yes. That's the way to go. But I'm very excited. And my background, I'm from Calcutta. So I'm just like you. I did my bachelor's, master's from University of Calcutta in statistics. And then I came to this country for my PhD. And after that, I'm a faculty at Clarkson University. I am very excited to have the interactive sessions on Sunday. That's my day. And I'm preparing for that. I have taught biostatistics in my school for a long time. And I have taught different other statistics courses, not just biostatistics. I think we can. Okay. I think somebody.
Hey. Tapia, I think you have to turn off your microphone. Otherwise, we are being interrupted here.
Please kindly off your microphone. Please off your microphone. we will just change into the webinar mode. After
Yes.
the inaugural session will be over. Otherwise, we have 600 participants, they cannot join. I will share a link over here. I will share a link in my chat box and you please join to that. Okay. And I do also request Shuman and other faculties also. So we are, but there is a little bit to say.
Papia, we were still listening to Shumana. Papia, oh, we are still listening to Shumana. Shumana, carry on. Shumana, carry on. Shumana, carry on.
Yeah.
Shumana, কিছু একটা problem হচ্ছে আমার এখানে slow হয়ে গেছে. Okay, you carry on.
Right. When I started using Zoom 2 years back, it happens all the time. Okay, so we are still learning the process. But really, I can see the faces, and I would like to see all of them on Sunday. And this is just a start, I would say. So we will continue our collaboration and interaction from now onward. And I visit India, Calcutta every year. So this is, as I'm saying, this is just a start. I'm ready to visit your college, university, and I would do that. And I'm from Ashutosh College and Baligan Science College. I have professors in every university in West Bengal. I have visited ISI, ISER, and then Vishwabharati, every university. So this is another collaboration that is added in my list. I'm extremely happy and delighted to be part of it. And I would like to see this program is flourishing. At the same time, I hope the students will get benefited out of it. I'm going to share my slides right after my talk, and I will have all my email address and every other details over there. So after that, if you have any question or anything that you'd like to know, definitely you should be reaching out to me and of course my graduate students. We all are here to collaborate and help others. And we know that statistics is integral part of every discipline these days. Without this subject, any research is incomplete. And again, I would like to thank Professor Ruma Bhattacharya for giving me this opportunity. And of course, I'm always thankful to Papia and Shujoy because they are my friends, as I'm saying, and I would really appreciate their effort doing this. It's not easy time. I mean, we all know that, and it's hard times, and hard times are teaching us hard things to learn. Thank you.
Thank you, Shumana. That was a very nice introduction, and thank you for your collaboration. You see, you are in New York. My younger one was in New York, just left for Michigan yesterday, 2 years. She was in New York Buffaloes, SUNY Buffaloes. So,
okay.
Yeah, so
close to my place, actually.
Your place? You did?
Yeah.
Oh, okay.
Like around 4 hours driving distance.
Oh, okay. So how is New York doing now?
New York is doing pretty good. New York is doing extremely good, actually. I think at least New York people are listening. So
that's something. That's something really important.
Yeah, we were the first hit state, but I think we are doing the best probably in US right now. But well, we'll see. Like the colleges, I mean, schools are still closed, the kids' school. The colleges are starting, universities and colleges. We'll see what happens after that. I mean, we cannot sit like this at home. Though we never stopped. It's not that we did stop anything. We have been teaching. We have been doing every other meeting. Everything. It was much busy time, actually, sitting in front of the computer. But that's not the way we want a university or a college system should run. We want interaction with students.
That's true.
We are, as you said, like when you go to your college and see the empty college, we never saw this in our life.
Yes,
and we don't want to see that in our life again.
Yeah, true. Okay, so thank you very much.
Yes.
Papia, to you.
Yes. Thank you, Madam. Thank you, Shumana. Actually, I am also monitoring the YouTube. There we have 21 participants and it's running fine. Now I would like Dr. Ghosh, Dr. Sukai Ghosh, Cytogenetics and Genomics Lab, Department of Zoology, University of Calcutta, to please come to the virtual screen and say a few words about this program because he is another collaborator of this program. So I would like, it's fine.
Okay, good afternoon to all. I want to congratulate Principal Vijay Krishna Girls College, all the teacher participants, the student participants, an organizer, and almost all who are attending this program right now. I am delighted and taking this opportunity to say a few words regarding these very unique and innovative programs that is initiated by Vijay Krishna Girls College under University of Calcutta. I hope this model of students training program and career guidance will cause particularly advancement in the student's career, and that will, I think, stimulate and energize the faculty member of other colleges to design such a program in near future, because it's a very innovative model. We have been talking about clustering of the college over the past few decades, but due to different types of logistic difficulties, we could not figure out this possible. But nowadays, this pandemic situation give us opportunity to think about the online clustering or virtual clustering of the different undergraduate colleges under University of Calcutta. And this is very essential under these circumstances, because different colleges under Calcutta University have different disciplines, and all these disciplines actually suffering from dearth of teaching resources. We have different colleges, and different disciplines of different colleges have different teachers of specializations, and that specialization and resources could be shared among the colleges through this kind of program, clustering program, and that would benefit the students to the great extent. Student would learn new thing. They may have some exposures in the different areas of their respective discipline in which they are studying. So again, I am congratulating the organizer, and I am happy and delighted to be the part of these organizations. And because this, what I am telling every time, that this is a beautiful model. We are testing this model, and if that will be succeeded to achieve student attention, then that could be implemented near future in the other colleges, and clustering will help to sharing the resources, human resources, knowledge resources to the great extent. Thank you all. We have several eminent resource persons from the national level as well as international level. Already Shumana has introduced herself. We have Shaunak Shahu, Dr. Shaunak Shahu online in this program right now. Shaunak is from NIH, National Institute of Health, Maryland, USA, and also from very eminent speaker like Budhaditya Mukherjee from Indian Institute of IIT Kharagpur, and so many others. Student, you will learn lot of things, lot of training, online training, and obviously will have some enlightenment regarding your future career. In what way you can shape your career, in what way you can thrive in the competitive life in near future, that will be enlightened by this eminent speaker. With these few words, I am taking opportunity to congratulate and to thank everyone once again. And now it's over to Dr. Papia.
Just a few words. Dr. Ghosh, I really welcome your support for our Department of Zoology of the college. Every time we are thankful to you that you have extended your hand to these kind of programs are, I would say, I've not so far seen this kind of programs happening anywhere else in any of the affiliated colleges. So we could be the pioneers in this kind of programs, and we are being able to do so because of your support of your department. So I thank you and welcome you.
Thank you, ma'am. Thank you once again.
Okay, so may I ask Indranil or Aditi or Abhinav, anybody here? Indranil, Aditi or anybody there? Okay. One thing I want to say that Shumana, I am going to introduce you officially, fully on Sunday as a person. Today you are a student and Sunday will be the resource person. Okay, I will introduce you to the students on Sunday. Okay, so now I have Shuman, are you there?
Yes, I'm there. I'm there.
Shuman, another Shuman.
Oh, I see.
Shuman, are you there? Shuman is not there. Hmm.
Dr. Shuman Bhushan Chakraborty from University of Calcutta. Have you joined the meeting?
Yes, we have
Dr. Shuman Bhushan Chakraborty.
Yeah, I'm there.
Can you hear me?
Please.
Yes,
Yeah.
of course.
This is your time. This is your time to start. So actually, from the end of organizer, I am requesting you to prepare yourself.
Shuman.
Okay. Please do introduce Shuman.
Okay, please off. Okay, can everybody hear me? Shuman, you keep your video on, please.
Yeah, I have on the system my video.
Shuman, I have one question to you. We have 100 participants over here and 162 participants on YouTube live. The problem is that today I need a meeting mode. As I have told you morning,
Okay, fine.
Meeting this in meeting mode. But in licensed version of Zoom meeting mode, 100 participants can be accommodated. But in webinar mode, we can accommodate 500 participants. So I want to know, should we shift to webinar mode, or for today we will continue in this particular mode, and in the YouTube we can have questions in the chat box so that we can actually discuss it with you. The rest of the students, 250 students, and who are not directly in the Zoom cannot interact with you directly, but we can pick their question from the chat box and can discuss with you. Tomorrow only we will shift to webinar mode. Okay, so it will be better.
I think we should continue in the meeting mode, because now shifting webinar mode would be a logistically hazardous. So stay online, students. Those who are in the YouTube link, please do attend the meeting and put all of your query in the YouTube chat box. We'll pick the questions. Professor Papia Ghosh will pick the questions from your end and will deliver to the speaker. Don't worry, all the questions and all the quests will be satisfied and answered. So we may continue in the YouTube in the meeting mode today, and from tomorrow onwards we will fix this program in the webinar mode. That will be better.
Yeah, I will send the bulk mail to all the participants by today night, just giving the webinar link. Okay, so today we will start with our first session, our first academic session with Dr. Shuman Bhushan Chakraborty. I would like to introduce Dr. Shuman Bhushan Chakraborty to all the participants. Shuman is actually very well known and very dear and near to me. He has done his graduation from Presidency College, Kolkata in Zoology in 1998 and has done his post-graduation from University of Calcutta as well as PhD from University of Calcutta. He has done his postdoctoral first class first position in MSc. He has received several national awards for his achievement in research related to aquaculture and fish biology, and his topic is also that he will be enriched by the knowledge of endocrinology and its application in the fishery, as well as he will also be enriched with the knowledge how to get jobs in fisheries and everything. He is engaged in the research work on reproductive endocrinology and stress physiology in fish model. Okay, he has presented his research work in various national and international conferences and published more than 60 research articles in various national as well as international, definitely peer-reviewed journals with high impact factor. So I am very happy to have Shuman with us, and I do welcome Shuman to this particular process, and start by sharing your screen. You can start, Shuman, by sharing your screen.
Okay, so Good evening, everybody. Am I audible?
Yes, you are audible, but probably due to rainfall or something, your net
Okay, fine.
You are audible.
Okay, fine. So in that case, very good evening to all, and thank you, Papia Ghosh, or rather Papia Di, I would say, for such a nice introduction of me, and of course, a very big thanks to the organizer of this International Student Development Program for inviting me to deliver a sort of interacting lecture with the students. So quite a long time back, I have just no longer teaching the UG students, or it would be a nice chance for me again to go back to this UG teaching mode or something. Maybe I'm just sharing my screen, and I'd be very happy to let you know that whether it has been shared or not. So, good. Yeah. Okay, the screen is being shared now.
Still not. এখনো তো দেখা যাচ্ছে না। হ্যাঁ, দেখাচ্ছে অবশ্য।
হ্যাঁ, এবার দেখা যাচ্ছে আস্তে আস্তে। শেয়ার কর। হ্যাঁ। Yeah.
Okay.
This is visible, Shuman.
Fine, done. Thank you, Shubhra. It's okay. So just like I have
presentation mode, Shuman. Presentation mode. Please put it in the presentation mode. It would be little bigger. Model. So often we are. So is it okay now? Yeah.
Fine. So I do work on endocrinology in fish model, and sometimes it becomes very difficult whenever we are talking about taking classes on endocrinology and then amalgamating endocrinology with fisheries. One is typically a classical or medical zoology portion, whereas the other one is typically a part of the applied portions of zoology. So I would just try to give a glimpse of how we can work on these two different aspects and how we can actually amalgamate these two things for future prospective orientation. So maybe at the end of my lecture, it would be clear, okay, what we can do after learning endocrinology and after learning fishery. But one thing we need to keep in mind that to make a very good career in fisheries, actually, it's better to be doing some bachelor's degree in fisheries itself, so the BFSC or the MFSC courses. But zoology as a basic subject is fine. I'll be discussing about all these opportunities from where we can go into line of fisheries after completing our BScs or MScs in zoology as such, as well as in terms of endocrinology. If we just recapitulate what we have in our syllabus with respect to BSc in terms of endocrinology and reproductive biology, in UG courses, actually, there are a diversification with respect to certain basic introduction, the hypothalamic hypophyseal axis, certain aspects of different peripheral endocrine glands, the few of the regulatory mechanism associated with these hormonal activities, and some non-mammalian vertebrates hormones as well. On the other hand, in the reproductive endocrinology, we have, of course, considering the male and female reproductive hormones and how the hormones are associated with reproductive health. Whenever, if a student, after completing his or her graduation, aspires to be a student of a master's in doing his MSc in zoology, he would be getting a chance to have a bigger picture of whatever he has or she has learned during his undergraduate courses. So it's basically based on whatever you're learning in the UG syllabus would be a bit more extended in the master's syllabus. So we would have again certain glimpses of this mechanistic orientation of hormone action. So in those cases, you would be finding, rather than only these two of cyclic AMP and IP3 DHA system, several other mechanistic orientations of signal transduction, a few more endocrinal glands would be introduced rather than only this thyroid, parathyroid, adrenal, pancreas, ovary, and testes. We'll be discussing about the details of pineal and other such glands, the gastrointestinal hormones and all these things, and we'll be discussing about more or less related to other functional activity of endocrinal orientation. And if we go for specialization in reproductive endocrinology, we'll be finding definitely a more of a reproductive endocrinological part associated with the reproductive health and the different orientation of male and female reproductions. No doubt about this. The fishery sector as well has different aspects associated with the whole part. Now, if we just go for a bit of recapitulation, I don't know whether all of you has gone through a certain extent of this UG syllabus or not. I just hope that the basic idea about what are hormones and how the hormones act, we all have this starting from the very basic level of class 9, 10 itself. We used to have our own ideas, or rather we like to prefer the hormone chapter in Madhyamik or Ucha Madhyamik because it was very easy to remember and it was also very scoring. We could just have a good score of these chapters as well. Endocrinology is nothing but study of hormones. So the next question is, what are hormones? So the hormones are, we all know that these are certain chemical or certain molecules which actually work to maintain physiological homeostasis. All the different metabolic activities in our body needs to be coordinated in a very fine way. It should be fine-tuned in order to maintain a very basal physiological level. Hormones does this particular thing. They coordinate the different physiological activities with each other to maintain a basal physiological level for our daily activities. So that is the most important aspect of hormonal functions. So they can be categorized chemically into different groups. They can be steroids, they can be peptides, they can be biogenic amines, they can be proteins or not. But the one basic thing is very much common in terms of any hormonal activity, that ultimately they are rendering a functional orientation which maintains a physiological homeostasis for any organism. So whenever we are dealing with these type of hormones and their functional orientation, the study is being known as endocrinology. So why the term endocrine has come? Because these hormones are often regarded to be synthesized by a very specific group of glandular structures or glandular system, which releases their content not via any ductular orientation or not via any ducts. Rather, they release them directly via diffusion into the system, and mostly they are being carried over through the process of diffusion into the blood vasculature system and being transported to the target organ. So if we just remember our very basic definition of hormone, we have often said that they are being released from one particular source and being carried by the blood vasculature system to a distant target organ. Now, that is not necessarily true for all the different types of hormonal functional activity. Sometimes we can categorize the functional orientation of a hormone in terms of autocrine, paracrine, or telecrine activity, where we can say that the hormones can be secreted from the source and acting on the same source. So the source and target remaining the same. So such type of function can be regarded to be autocrine. Whereas if the hormone is just acting over a very short distance and simply diffused to a very closely associated target organ, then it becomes a paracrine activity. But the truly telecrine or endocrine activity are those where the hormones are secreted by one particular source, being carried through the blood vasculature system and ultimately reaches a distant target organ for its functional orientation. So then it becomes a telecrine activity, to be more precise, and we call them to be the endocrinal functional orientation. But the term endocrine has actually come from the system where the material, the chemical, is being secreted by a gland without any duct. Then, so as we were saying that they are reaching to a target and then eliciting their function. So there must be an interaction between the molecule with its corresponding target. So that interaction is mediated by a particular group or different group of receptors. So any hormone is functionally acting via the receptors. So the receptors can, again, depending on what type of hormone it is, can be of different two principal categories. One group of receptors are actually located over the plasma membrane itself, the membrane-bound receptors, whereas the other group of receptors are located within the cytosol or within the nucleus. So depending on the chemical nature of the hormone, the receptor also varies. The hormone can actually bind with the membrane-bound receptor, or can bind with the, or can just simply pass through the plasma membrane and enters within the cytosol and bind with the cytosolic or nuclear receptors. Once the receptor ligand binding is there, subsequently there can be different mode of functional orientation, either in terms of a signal transduction mechanism altering different enzymatic or biochemical activities, or they can the hormone receptor complex can actually act as a definite transcription factor and bind with the potential responsive element over the promoter region of a target gene and alter the transcriptional rate of the target gene, either upregulating or downregulating production of mRNA and ultimately leading to production or down production of different type of proteins. So by this way, the hormone can act either altering the biochemical activities, directly affecting the enzymatic functional orientation, or the genetic activity by producing the product or by altering the rate of production of different genes. In either way, they are manipulating the total system in such a way that ultimately, with respect to the functional aspect of any hormone, the physiological level. So that's a very basic orientation for any hormonal activity. So you all know that the lipid hormones, the steroid hormones, they can simply pass through the lipid bilayer, and mostly the steroid hormones, their receptors are cytosolic in nature, whereas the protein hormones or the peptide hormones, they cannot pass through the plasma membrane. So they require binding with different types of membrane-bound receptors to have their functional activity via the second messenger system. Now, these sort of basic things would be dealt with in the BSc syllabus, of which two have been mentioned in the syllabus very specifically, the cyclic AMP system and the IP3-DHA system. The rest, like the JAK-STAT pathway, there are several other the toll receptor related pathways and different other signal transduction mechanism will be dealt with in master's courses. Now, just we can look into a very basic aspect of any hormonal activity. This is a very basic regulatory orientation. As we are saying that the hormones regulate different functional orientation of the body. So can we say that all the hormones are actually having very much well regulatory functional orientation? So the hypothalamo-hypophyseal axis is actually the master regulator of this entire functional orientation. So you can just have a glimpse of this structural organization where we can consider the hypothalamus to be the boss. There are several neurosecretory cells being present over the hypothalamus, which releases different types of stimulatory or inhibitory factors or inhibitory hormones, which control the functional activity of the manager, that is the pituitary gland. So often we are supposed to know that, okay, pituitary is the master gland, regulating the functional activity of any tropic endocrine glands and regulating secretion of different endocrine glands. But there is a master over the master, that is hypothalamus. So the neurosecretory cells of hypothalamus, they are releasing different types of inhibitory or releasing factors, ultimately regulating the functional activity of pituitary as well. So you can just have an example with respect to this TSH system, the thyroidal axis actually. So you can just call it as the hypothalamo-hypophyseal thyroidal axis as an example. All the peripheral glands, they have this type of regulatory mechanism associated with pituitary and hypothalamus. So the boss, the hypothalamus, they are releasing TSH releasing hormone or thyroid stimulating hormone releasing hormone, which in turn is affecting the pituitary gland to produce TSH, that is a thyroid stimulating hormone. In turn, the thyroid stimulating hormone is regulating the worker, that is the thyroid, for production of thyroxine, T3 and T4. So in this way, the basic mechanistic orientation of hormone works. Of course, there is a feedback system with respect to this type of peripheral functional hormonal activity, where secretion of thyroxine in turn can have a feedback, negative or positive feedback for that mechanism over both the secretion from pituitary as well as from secretion from hypothalamus. We call it to be either via a very short loop system or via a long loop system. So if we just have, we can again say the same example, where if thyroxine regulates the functional orientation of pituitary secretion of TSH, then it becomes a short loop system. And when thyroxine is regulating the secretion of TSH RH from hypothalamus, so that is Walker is regulating in the boss rather than bypassing the manager, so then it becomes a long loop system. And such type of functional feedback mechanism is definitely being present with respect to any endocrinal functional orientation. So any endocrine hormone is actually having such type of feedback mechanism. And as we have talked about the different types of receptors being present, so there is one very intricate relationship between the hormone with its corresponding receptor. Often it has been found that a particular hormone is binding with a very specific group of receptor depending on the structural compatibility. So the hormone's binding with a particular receptor. It's just like the enzyme substrate activity. There is a structural compatibility with each other. But is it true that always the one hormone is going to bind with a very single specific receptor? Sometimes that doesn't happen. There is a group of receptor where a single hormone can actually bind with a more than one similar type of receptor. And the converse is also true, where the same receptor is actually binding with more than one hormone. But we must remember that because of this structural compatibility, the functional orientation varies with respect to such binding. So again, we can take an example. If we consider one hormone A has a very specific receptor in terms of receptor A1 and A2, and the structural binding propensity of hormone A with A1 is more than A2, then of course, when A is binding with A1, the functional activity would be more compared to A binding with A2. So that means if we have one molecule of hormone and it is binding with one molecule of A1, the extent of function would be more rather than one molecule of hormone binding with one molecule of A2, or we may say, in order to give the same extent of functional orientation, one molecule of or other A2 has to bind with more molecules of hormone. So that means the concentration requirement for hormone would change with respect to the type of receptor it is binding with. And this particular concentration of the hormone is being known as EC50. We call it as effective concentration 50. So what I'm trying to say is like, the concentration which is required to elicit half of the maximum functional activity is being known as EC50 for a hormone. So of course, lower the EC50, the better is the functional orientation between the hormone with its corresponding receptor. So if hormone A is having EC50 of 1 with respect to receptor A1, and hormone A is having EC50 of 2 with respect to receptor A2, we can say that the binding propensity of A with A1 is better compared to that of A with A2, because the EC50 is lower for binding between A and A1. So in this way, we can have different dynamics with respect to hormone functional activity as well, and those would be dealt with in advanced studies as well. And sometimes we may find that there is a threshold level of hormone receptor binding for eliciting when with one molecule of receptor to elicit the maximum functional effect. That is not always true. It may happen that minimum 10 molecules of the hormone should bind with 10 molecules of receptor to elicit the maximal functional activity. So whenever that happens, the minimum number of receptor molecule which is required to bind with a hormone to elicit any function, that has been known as threshold receptor occupancy. So these two terminologies are very much important in terms of the dynamics of hormone function, the EC50 values and the threshold receptor occupancy values, where that is the minimum number of receptors requirement are required for eliciting any functional activity of a hormone. So if that happens, the number of receptor over a target is actually more than what is required for this threshold receptor occupancy, then we call it to be having some sort of spare receptors, just like the spare parts in any toolbox or in any car. So the spare part, the functional activity of spare part is if somehow the existing working parts are lost or is not functioning properly, we can have a mechanism by which we can substitute this existing part. So those are the importance of spare parts. So that's the same for spare receptors as well. The spare receptors are actually those which are in excess of the minimum requirement from the functional activity. So obviously, if some target cell is having a number of spare receptors or more number of spare receptors, it is better for the target to be more reciprocating against the hormone rather than one cell or one target which don't have any spare receptor. We can simply have an example with this respect as well. Say for example, one target is having 10 number of receptors, and in order to elicit the functional activity, hormones should bind with all 10 receptors to elicit the function. So if the target cell is having 10 number of receptors and somehow one receptor is lost, what would happen? Even if there is a huge number of hormone concentration or if there is a sufficient amount of hormone being present, in absence of 10 receptors, no functional activity, or rather the maximum functional activity can never be achieved. So we cannot have the maximum functional potency if the number of receptor is reduced once there is no spare receptor. But think in a different way in another target when there is 15 receptors being present and we require 10 receptor to elicit functional activity. And you reduce the number of receptors from 15 to 12. Still, we have more number of receptors to elicit function. So that means obviously we can achieve maximum functional potency even if the number of receptors are reduced if we have spare receptors. That's the importance of having spare receptor in a system. But of course, in that case, what would happen to the functional orientation? The EC50 will increase, because if we just look into simple mathematical orientation, if we have 15 number of receptors and we require 10 for binding with hormone to elicit maximum function, the probability that 10 molecules of hormone will bind with 10 number of receptors when there is 15 receptors is obviously more compared to if there is 12 number of receptors. So in that case, EC50 will simply increase, but still we'll be achieving the maximum functional orientation with this sort of receptors. So these are now, this is a very basic, we all know this, we are studying this from our very class 9-10 itself, the major endocrine glands. Besides these major endocrine glands, most of these glands would be dealing with in the BSc syllabus, and as I have said, the pineal gland would be dealing in the advanced MSc course. Then there is this thymus, which would be again dealt with in MSc, and besides these preliminary glands, we would be dealing with the gastrointestinal diffused system of endocrinal activity in the MSc courses as well. Now, just coming to what we can do after going through these endocrinological studies. So we can categorize the studies in endocrinology into these three major branches. There can be several subgroups. I have just for a very basic orientation have divided this into three. The main aspect is related to either reproductive endocrinology, neuroendocrinology, or clinical endocrinology. So coming one by one, what's the importance of reproductive endocrinology? We all know that. So this is basically a specialized type of endocrinological orientation where we are dealing with hormonal functioning with respect to reproduction. So mainly the hypothalamo-hypophyseal gonadal axis. In case of male, it would be the hypothalamo-hypophyseal testicular axis. For female, it will be hypothalamo-hypophyseal ovarian axis. But in both the cases, often we are finding that there are several difficulties with the advancement of lifestyle or with the change in lifestyle. There are several diseases, that cases of infertility and all these things have increased. The reproductive problems in couples have increased with the change in the environment, with change in our lifestyles, and often the endocrinological orientation of these diseases are very much important. So that's particular part of endocrinological study is being branched into reproductive endocrinological part or reproductive endocrinological aspect. In addition to the medical aspect orientation with respect to infertility, is hormonal or reproductive endocrinology is also important with respect to production in terms of increasing demand of animal breeding, be it fish, be it dairy animals. Often we resort to this type of hormonal manipulation with respect to increasing productivity. We want to produce a good breed of cattle, or we want to produce a better growth orientation in terms of fishes. So we go for such type of reproductive manipulation with endocrinal manipulation in terms of reproductive endocrinology. Neuroendocrinology. Now, I'll be coming into this again later. It is basically the study of interaction between nervous system and endocrine system. Now, these nervous system and endocrine system are actually two parts of the same thing. We cannot altogether differentiate endocrinology from the neural system because they are very closely integrated with each other, and that integration comes from hypothalamus. So often it's been done that the several types of neural impulses, the several stimulus, be it light, be it touch, or different other types of chemical stimuluses, they're often being perceived and analyzed in hypothalamus, and accordingly, the hypothalamus starts secretion of different neurosecretory substances, which in turn regulates the functional activity of pituitary and ultimately production of different peripheral hormones. So these very much integrated orientation of the neural system. So often we are finding, you can just cite one example with respect to our activity of adrenal medulla. So you all know that adrenal medullary hormones, the catecholamines, the adrenaline and adrenaline, are noradrenaline, are often being termed as impulsive stress hormones. So immediate stress is there and we react with respect to adrenaline. We are all familiar with the terminology of adrenaline rush. So what happens? We are just going for a very exciting ride with bike ride or something like that, and we feel a bit of more secretion of adrenaline. We are going for an exam and there is an increased secretion of adrenaline which keeps us prepared or makes us alert for such type of impending stress in terms of examination. We're moving in a dark alley and all the senses are becoming more active, more alert because of the ambience. So all these neural stimulations are ultimately integrated with respect to hormonal secretion of catecholamines. So this is the functional orientation of neuroendocrinology, that the integration between the neural stimulus with that of endocrinal secretion ultimately affecting our physiological status. So whenever Virat Kohli goes on playing Pakistan, he would be having a more secretion of adrenaline rather than whenever he is going to play Afghanistan, I would say. So rather just because it's a more high voltage match, so the stakes are more. So of course there would be a bit more alertness would be coming into everybody, and that would go on for increasing the secretion of adrenaline. That ultimately enables us, that ultimately makes our senses more proactive, and we perform better with respect to secretion of this adrenaline. Okay. So that's the importance of having this type of neuroendocrinal integration system, which affects our cognitive and behavioral functioning. So a fielding at the slip is cognitively more alert compared to that of a fielder who is fielding at the outside outfield. So that's simply because the fielder at the slip knows that there would be a very much importance of greater reflex action rather than at the outward fielding, who has comparatively has a higher time to see the ball and to react to the movement of the ball and all this. So that's the importance of cognitive behavior, and that's the importance of secretion or integration between the neural system with that of endocrinological system. So the endocrinal function, which have a very strong foundation with respect to central nervous system, and as we have said, that's why we are always talking about this hypothalamo-hypophyseal axis, that the hypothalamus secreting different type of hormones are ultimately regulating the secretion from pituitary, which in turn is regulating secretion of different peripheral glands. The pineal gland is another important issue in these cases because this pineal gland is often being associated with our circadian rhythmicity. We all know that we are diurnal in nature. So we sleep at night and we wake up in the morning whenever the sun rises. So we have some basic knowledge related to this such type of circadian rhythm, where we call this simple process to be known as the Zeitgeber. So the Zeitgeber is actually the light that that entrains our internal clock system, our internal mechanistic clock, to be associated with the external stimulus. So if somebody is having a problem with respect to this entrainment of this external stimulus with that of internal clock system, then it becomes a problem. We often call it about jet lag. If someone of you has an experience of just moving to America or coming back from America to here, we all feel a bit of dizzy during the daytime, and we don't go for sleep during the night. Sort of amnesia is there. So simply we don't think, okay, we are not having sleep at night and just feeling sleepy during the daytime, simply because our system, our internal clock mechanism associated with this hypothalamus and the pineal gland are oriented in such a way that we entrain them with respect to sunlight, that okay, this is supposed to be during the daytime, so I must be active. So there is a very strong correlation of all this pulsatile secretion of different hormones like cortisol, which is secreted more during the daytime and less during the night. So all these things are very much well integrated in the neuroendocrinological aspect of our syllabus. Then the clinical endocrinology, and that is of course one very important aspect, one very important part of research in nowadays. But this is a field of medicine that diagnoses and treats diseases of the endocrine organs. So you all know about this diabetes, and people are during the time of COVID, uh, the most rather talked about term is comorbidity. The mortality rate is higher for those who are suffering from diabetes. So diabetes is definitely, we all know that it's an endocrinal disease. So clinical endocrinology becomes very important in all these aspects. The different thyroid disorders, the metabolic disorders, obesity, again a lifestyle problem related to endocrinological orientations. Endocrine tumor syndromes, the disorders in childhood and adolescence, very much problems with respect to development of puberty, the adolescence problems, the bone and mineral metabolism. So several, we have all seen the advertisement of women's horlicks or the protein horlicks for those, which is saying that okay, after a certain period of age, the mineral density in bones get decreased. So we need to have compensate those things, and those are related to different endocrinological aspect. With after menopause, there is a decrease in level of estrogen. So once estrogen level is low, the ability of calcium retention by the bones decreases and bone mineral density is being hampered. So all these parts are being talked about in the topic of clinical endocrinology. So now coming to what we can do after studying this endocrinological part. Okay, fine. Some of us are going to go for higher studies. So after completing BSc, I feel interested in doing MSc, and then during the MSc time, I thought, okay, fine, I'd be very much interested in doing work on endocrinological aspect. So definitely we can go for NET, GET, or giving GRE. So the University of Institutional Entrance, different types of entrance examination, but be very sure that these are not actually some exams which will enable you or which will ensure that you will be getting jobs. So that's a very difficult proposition, I would say. To get jobs, what should we do? But anyway, if we want to do higher study, for higher study, these are the exams that must be cleared. Unless and until you I have either cleared NET or GET or GRE. GRE is for if you want to go abroad to do your MS and then do your PhD from USA, then it's needed. But if you want to do it in India, NET is the most important, followed by GET IST. So you can go for doing your PhD, and from there you can look for doing your postdoctoral studies, become a scientist in any institute, or become a professor in different universities or colleges for that matter. On the other hand, if we just look, and for those people who are wanting to do higher advanced studies with respect to endocrinological things. So as we have said, there are so many problems being associated with endocrinological things. We can look for these neurodegenerative diseases. So we all know that the extent of neurodegenerative diseases, previously we all know amnesia was there. Okay, we are forgetting something, but consider this Alzheimer's. Now the disease has come into foray. Okay, with age, our cognitive system is decreasing. We cannot memorize all these things. So gradually our brain cells are drying. Is there any endocrinal aspect with respect to this type of diseases? That's a very important and pertinent questions now. We are all know about Irfan Khan being dying out of neuroendocrine tumor. So once a celebrity is being affected with that disease, the disease come into, I would say, in the newspaper. So of course, people who are working with endocrinological research, who are engaged in endocrinological research, they knew about this sort of tumor which can happen in the neuroendocrine system. These are growth of cells that actually manufacture hormone, which regulates body's functions. It can be associated with our pituitary tumor, can be associated with tumor in the brain regulating or malfunctioning of the hypothalamus. So all different types of these tumors can be there. There can be pancreatic tumors as well. So endocrinal gland tumors are often being a good issue of research work. Then infertility problem, as I have told earlier, that nowadays more and more couples are facing such a problem with respect to infertility. Maybe what are the reasons behind that? The different types of increase in pollutions. All these polluting reagents can actually act as endocrine disrupting chemicals. Different types of EDCs, ultimately affecting the ability of a person to produce the different reproductive hormones and ultimately produce functional sperm and ovum. So the association between different types of such EDCs and how it is affecting our reproductive capability. So many problems to ponder over. Then the increase, a gradual increase in diabetic patients in India. So as we are seeing that, previously it was thought, okay, diabetes that's going to happen after say around 50 or 60 years of age. But now we are finding that even there is child diabetes as well is happening, and people are in a child of as minimum as 7-8 years of age, they are also suffering. Sometimes the news are coming out, okay, they are suffering from diabetic orientation. And even at the middle age people, the people above 30, they are also suffering from different stages of these endocrinal problems. So why these things are happening, why such endocrinal problems are actually occurring in different societies? So those are can be, or rather those can be the topic of research for you people. So here is a list of some of the research institutes where we can, of course these are all foreign universities mostly, where we can go for working in endocrinological studies. Okay, it's a ranking is being there, mostly associated with endocrinology and metabolism. But what I'm trying to say is, if we want to go for advanced research work in the field of endocrinology, there are ample scopes. But what you need to do is you have to have a very well grasp of basic endocrinological knowledge, and you want to think out of the box, or rather you have to think out of the box. You have to think of a problem which is quite pertinent, quite relevant in terms of modern society, as well as not many people are working on that. So that has to be done by the particular person himself or herself. And if you don't want to be that much of a very advanced researcher, I don't want to go for any further research work in terms of endocrinology. But with my endocrinological knowledge, can I do something? Do I have any basic job proposition for this sort of things? Okay, often this type of works are mostly being occupied by the people who are trained in such type of uh, technologies. Like endocrinology as a special orientation is being done in medical practices as well. So we can often find a situation somewhere, a person who is a doctor is specialized in endocrinology. So those endocrinological doctors, they can do MD in endocrinology and all this, which is beyond the scope of this particular lecture, because we all know that we are going to do BSc in zoology and then look for certain jobs. So what we can do is, like, we can go for certain diploma courses associated with this hospital technology management system. So some colleges are there which offer this sort of diploma courses in hospital management system or technical aspects of doing with different instruments and all this. And in those cases, if you have a very basic knowledge of doing different what should I say, different techniques associated like RIA or ELISA. So all you have a basic knowledge of doing these type of things, which are very much part and parcel of a job of a technician in a pathological lab. Then we have these sort of job orientation or job opportunities as well. So they can work in pathological laboratories or in clinics. As I have said, they must have some basic diploma degree in these aspects, like the nutritional therapies, certain extramural training rotations or training courses are being offered. We can go for medical auditing, the record keeping, and we have to know the hormone assay procedures. Okay. So like the RIA, ELISA, the Western blotting system, all these systems, all this technical knowledge will give us a chance to be absorbed in the field of the newly advanced stages of hospitality or hospital management system. Okay, as I have told that I will try to integrate between fisheries with that of endocrinology. I'm now coming to the part of fisheries. So, okay, fishes being a vertebrate, of course, they do have endocrine glands. But the endocrine glands in fishes, they are more or less same to that of mammals, if we consider human. So there are some similarities between the human endocrinal system with that of the fishes, and of course there are some dissimilarities as well. So now often it happens the fishes, they have few discrete endocrine glands like the pituitaries, the thyroid, and the pineal. These glands in fishes are typical discrete type of endocrine glands, whereas they also have certain glands which contain both endocrine and exocrine functions. So some glands are there which are dual in nature. So these dual functioning glands like the kidneys, the gonads, and the intestine, they contain or they perform both the exocrine part and the endocrine part. And often there is an interesting orientation with respect to fish endocrinological aspects. Like the thyroid, I've told you that thyroid is a typical endocrine gland, is a discrete gland, but there is often presence of heterotopic thyroid follicle in the kidney itself. So in certain cases within the head kidney or in heart even, certain thyroidal tissues can be found in fishes. Now, how do we define a thyroidal tissue? We define a thyroidal tissue by virtue of its ability to incorporate iodine and produce iodinated amines. So if some tissues except the specific thyroid gland has the ability of incorporating iodine and then producing iodinated biogenic amines or iodinated amines for that matter, we'll be saying that, okay, they are similar to thyroid. So in fishes, such type of heterotopic thyroid glands are being found in heart, in intrarenal system, in kidneys, and in head kidney region. Besides that, they also have scattered cells with endocrine function, which are also being known as diffused neuroendocrine system or diffused neuroendocrines, mostly being present in the interdigestive tract of fish. And often they are acting in a paracrine way. That means they are being secreted from a particular gland or particular region in a diffused glandular system and acting in a very nearby target system. And if we look into the chemical nature of these fish hormones, of course, just like the mammalian counterpart, they can be categorized into steroids, peptide or proteins, and the amino acid analogs. So these are the different endocrine glands which are being present in fishes actually. So as you can see, the pituitary, pineal, hypothalamus are the common one. The thyroid is there. Then the gonads, all these things are the pancreas. Okay, these are fine. But besides those things, the intestinal mucosa can also act as an endocrine system, and few additional structural organizations like corpuscle subcutaneous, the urophysis, and the ultimobranchial body. These are very much distinct type of endocrinal glands or endocrinal tissues which are being present in fishes, not being found in correspondingly in mammalian system. So I'd be just taking one into one. As you can just see here, the section of pituitary, and the pituitary in typical fishes can actually be having these gross parts, the adenohypophysis or the anterior and the neurohypophysis. The adenohypophysis again being divided into proximal pars distalis, rostral pars distalis, and pars intermedia, and secreting different type of hormones which are more or less same like that of mammalian counterpart. But one thing is important here, that there's a pars intermedia, which is secreting melanophore dispersing hormone and melanophore contracting hormone, are very much important in fish, because in mammalian system we all know that, okay, our pigmentation pattern is not that one fine morning you are, okay, we are rather brown in color, I would say, and one fine morning I just get up to find out to be just very black in color. That won't happen for us. Or very fair in color. That on the overnight we cannot change our body color and our pigmentation pattern, the deposition pattern of melanin. But consider a fish. They are actually, there are several, the coral fishes are there which can change their coloration pattern just by changing the orientations of these pigment molecules being present within the cells by manipulating the secretion of melanocyte, this dispersing, and the melanophore contracting hormones, regulating the orientations of these melanophores within the system, within the skin, and thereby regulating their body color pattern. So this is very much important functional activity. For us, this melanocyte stimulating or melanophore stimulating hormone or MSH is definitely important, but not that much of like that of a fish. And coming to this pars neurohypophysis or pars nervosa secretion of vasopressin and oxytocin. So you all know that the secretion of vasopressin and oxytocin is very much important in us, in fishes as well. These two neurohormonal peptides, as you all know, are being secreted by the hypothalamus and being stored in the posterior pituitary. So they are basically, the posterior pituitary organ is acting like a storage release organ. The hormones secreted from the hypothalamus getting stored in the posterior pituitary and being released in due course. So this mammalian oxytocin and vasopressin have similar counterpart in fishes. If we consider the agnatherms, they have only one type of these posterior pituitary hormones. Whereas the most fishes are actually secreting an oxytocin-like peptide, which is also being called as isotocin. Okay. So most of the fishes are actually secreting isotocin, which is actually a type of oxytocin-like peptide. And the second one, rather than vasopressin, they secrete arginine vasotocin. So this arginine vasotocin is actually a hybrid of oxytocin and vasopressin. So their chemical structure is such that it contains some functional properties of oxytocin as well as some properties of vasopressin. So just like this arginine vasotocin can actually function like oxytocin, stimulating contraction of muscles of the reproductive tract, leading to or playing a role in egg laying or in birth system, and they can also act like vasopressin with either antidiuretic or diuretic properties. So these are the important differences with respect to the functional activity of pituitary in fishes. Coming to thyroid gland, we all know that the thyroid glands in fishes are typically like that of the mammalian system. They can be located in bony fishes. They are actually lying under the first branchial arch on each side. But it is found can also be found along the afferent branchial arteries of the gills as well. And the follicles, as we have told, they can be heterotopic as well, migrating to liver, kidney, brain, and different locations altogether. And they're chiefly secreting thyroxine, regulating metabolism, oxygen consumption, then to have some role to play influencing osmoregulation and influencing migration of fishes. Now that would be coming into later on. Then the adrenal gland. Okay. So now this adrenal gland or the internal gland is a bit different from that of ours. So in mammalian system, we all know that there is a definite adrenal gland above the kidney, and this adrenal gland encompasses both the cortical tissue and the medullary tissues. But here what we are finding for fishes, the two components, the cortex and the medulla, are actually separated. So often there is aggregation and distribution of the adrenal, this adrenal tissue varies in between different teleostean fishes. The steroidogenic cells, that means the cortical cells, they are aggregating towards the head kidney region. So this is the head kidney part where the corticotrophic cells are being localized. And often in between these corticotrophic cells are interspersed the chromaffin cells. Okay. So just beneath these cortical cells, the medullary tissues, the chromaffin cells are being localized here. So a more or less diffused pattern rather than a very concrete orientation of cortex and medulla. What we'll be finding in fish is a diffused orientation of these cortical cells and a separated organization of these cortical cells and the medullary cells. But functionally, the adrenal cortex are definitely steroidogenic, producing the adrenocorticoids and the mineralocorticoids, which help in the process of osmoregulation, protein and carbohydrate metabolism, and they also help in blood cell movement from hematopoietic and lymphatic tissues and growth. Whereas for this adrenaline, they are mainly involved in the process of metabolism. Now coming to a specialized structure in fish, which is known as corpuscles of Stanius. So these are often embedded within the kidneys in fish, releasing a particular proteinaceous substance, often being called as hypocalcin. The functional orientation of this hypocalcin is to regulate calcium balance. Now, this ionic balance in fishes are very much important. They are living in an aquatic environment, which can again change with respect to if they are migrating from one particular environment to the other. Then with respect to that, they have to regulate their ionic balance within the body. So in those cases, there are so many hormones being involved in ionic regulation in fishes. Like this hypocalcin is one of them. I've talked about the mineralocorticoids, again associated with this type of ionic balance. Thyroid plays an important role, and coming later, prolactin is another very important hormone associated with maintenance of ionic balance in fishes. Then after this corpuscles of Stanius, there is ultimobranchial body, which is another specialized type of cells or specialized type of endocrinal glands, which are sac-like structure between the ventral wall of esophagus and the sinus venosus, again releasing calcitonin, which is associated with calcium maintenance of calcium level in blood. And finally, there is urophysis or the caudal neurosecretion being present, located close to the tail of the teleost. So they are mainly slow swelling type of glandular structure being present at the posterior or caudal end of the spinal cord. And they are secreting different type of urotensins like urotensin 1, 2, 3, and 4, mostly associated with metabolic regulations. Like urotensin 1, which is related to regulation of blood pressure, they increases blood pressure. 2 is involved in contraction of smooth muscles of urinary bladder. 3 is associated with sodium intake across the gills, and 4, activity like antidiuretic hormone of pituitary gland. So a variety of functions are being done by these urophysial secretions. So they secrete different type of hormones which are associated with different type of functional orientations. And finally, this is a figure of pancreatic islets being present in fish, which is again involved in the process of carbohydrate metabolism, just like ours. So the gut walls of the hepatopancreatic structures are being present. Most bony fishes, they have this hepatopancreatic structure where a particular portion of the pancreatic tissue are amalgamated or associated with liver. So there is a very close degree of association between the liver structure and the pancreatic structure. So these are the pancreatic islet structure which are being present, the PL1s. So often these pancreatic islet cells are being closely associated with liver, and they take part in carbohydrate metabolism, and mostly they secrete insulin. Now coming to the very important orientation of how to correlate with this endocrinology with fisheries. As the title of my topic was like integrating fishery endocrinologies with fisheries. So how we are integrating these two? If we just look for the fisheries orientation, we can broadly categorize fishery into these two aspects, the fruit fish culture and the ornamental fish culture. So often, culture fishery is being oriented with respect to terms of production. Either we are going to produce fish which we are going to consume, or we are going to produce fish which has got some sort of aesthetic value. So we are keeping them in aquarium, looking at them and finding ourselves happy. Okay, fine, they look so good. So whenever we are talking about food fish culture, the main objective in these cases are either to enhance breeding, so we can always go for developing technologies related to breeding. We are often looking for increased production, so definitely we are looking for increased growth. So if you just go to the market and find one fish size of about 500 gram and another one of size of about 1 kg, and talk about hilsa, and we would definitely think of, okay, 1 kg hilsa would be better compared to 500 gram hilsa. So of course we'd be looking for growth in fish. Then immunity and stress. So many issues are there. We are culturing the fishes under a very highly intensive system with a very great number of increased stocking density. So whenever we are confining them in a very small space, we are immunocompromising them. We are providing them stress. The fish are being transported from one place. The live fishes are transported from one place to a very distant place for marketing. So all these are providing them with stress. We are never going to buy some fish in the market which are already dead. So we always look for, okay, whether the fish is alive again. বাংলাতে বলতে পারি যে খাবি খাচ্ছে কিনা মাছগুলো এখনো। So whatever happens with these things, we want to find out, okay, whether the fishes are healthy or not. So definitely the producers will try to increase or to go for a betterment of immunity, and so that the fishes are able to withstand better handling stress. And then there is the process of migration. We all know about, we are talking about hilsa, and the major aspect of hilsa is their migration behavior. So they are migrating from the sea to the riverine system for breeding, and then after breeding they're going back to the seas again. And during such migration, we can often say the taste of the fishes are actually oriented with the process of this migration on what stimulates them for such migration. If the hilsa don't migrate, so what would happen? So all these can be related or can be integrated with the endocrinological aspect as well. We're coming into that. The breeding, growth, the immunity stress, the migration, everything of fisheries which are associated with food fish culture, so it are being related to the endocrinological physiology of the fishes. So I've just give the logo of this International Society for Fish Endocrinology. So it's not like I'm doing something which is out of the box or beyond the scope. Okay, I have done my PhD in fisheries, but I was a student of endocrinology special paper, so I have integrated. No, it's not at all like that. A branch of science is definitely there who deals with combining fisheries with endocrinology. So it's there is always an option that you can combine these two. For ornamental fish culture, of course, we are wanting to buy something with more color. So color enhancement is definitely an issue with respect to ornamental fish culture. And as we have said, the color pattern in a fish often depends on the distributional pattern of the melanophores. And the melanophores are being regulated by this MSH. Hello?
Sir, good evening.
Hello?
Mute yourself.
Pardon?
Otherwise, I will remove you. Who is this?
Sir, please continue.
Otherwise, I will see and remove you. After the completion of the session, sir will interact with you. So if you are not interested, you can just get out of the particular meeting. And if you are interested, then mute yourself. I can find who is doing this.
Sir.
Shuman,
Sir.
Okay, so we can always look for endocrinal orientation of this color enhancement in ornamental fish culture. Okay, and as we have said, immunity and stress is often an issue with respect to transportation of fishes, and we all know that these ornamental fishes are often being transported quite a long distance from one country to the other countries, and there are several exporting markets for these type of ornamental fishes, where we must send them in a very live condition. So for this,
ऐसे हुआ मेरी
Shuman, you continue. I will find this particular participant and see what I can do.
Sir, please continue.
Please continue. I am finding this participant who is unmuted right now. Okay, I am finding out. Just a minute, Shuman. Moni, you please go through the participant.
Yes, yes, ma'am. Yes, yes, ma'am. Yes, ma'am. More than 3 to 4 participants are unmuted condition.
Remove them. Just remove them. Remove them. Just remove them.
Okay, ma'am. Okay, ma'am.
I'm also doing. I'm also finding.
Okay, ma'am. I think we have removed all of them.
Yes. Anjana Sinha.
You can start, Shuman. Sorry for the inconvenience. This is the problem of meeting board. You can start, Shuman.
করাচ্ছে তো
Shuman, are you there?
Sir,
Shuman?
please start.
Shuman, can you hear me?
Sir, তো unmute হয়ে গেছেন.
Sir, please, yeah, Shuman, please unmute yourself. Shuman, please unmute yourself. সুমন কি নামে আছে একটুখানি
স্যার নাম্বার আছেন 8124 ওই নাম্বারটা হচ্ছে স্যারের,
কিন্তু এটা আনমিউটই আছে। সুমন আনমিউটই আছে, সুমন আনমিউটেডই আছে।
Okay, can you hear me now?
Yes sir.
Okay, thank you.
Okay, fine. So,
we have removed. Moni, continuously watch. Okay, I am also watching.
Okay ma'am, I'm watching.
Whoever is unmute, throw a name, just point the name.
Okay, so should I start again?
Come on, absolutely. Carry on kindly.
Okay, fine. Okay. So whenever there is certain environmental cues like change in day length or change in REM pattern, or all this gives a neural stimulus to the process of reproduction in fishes. So what happens normally, as you can see here, that dopamine is a negative factor associated with reproduction, whereas GnRH, the gonadotropin releasing hormone coming from hypothalamus, that's a positive factor. So any neural stimulus associated with reproduction would do to reduce dopamine secretion by the hypothalamus and increase GnRH productions. Now, so there are several integrating pathways associated with all this process. Okay, this within this short period of time, I can just give a glimpse of whatever we are going to study during MSc and during the higher level of research. We'll be finding that in the hypothalamic region, there is a typical interrelationship between this dopamine, there's GnRH, and one other factor known as kisspeptin. And all these three things act in a synergistic way, where when if some fishes want to go for reproductive stages, there would be a reduction in dopamine secretion with an increase in GnRH production, which in turn will regulate the pituitary if it is during the growth phase of development to secrete follicle stimulating hormone, or FSH. Now in fishes, often it happens the GTH or gonadotropic hormones are being regarded to be a single one, as there are so many different species of fish and works has been done with different group of fish species, and it has been found that some of the fishes are actually having a single type of analog for GTH, which is being known as GTH-A, which is mostly like that of luteinizing hormone. So the role of FSH in fish reproduction is often being debated in such a way that some of the fishes, they have a good role of FSH, and some there is not that much of significant role for FSH, rather, but LH is having a more important issues associated with the entire process of fish reproduction, of course. So whenever we are dealing with this type of breeding experimentation, I try to produce something which is more of an analog to LH. So often we are calling it as LHRHA or LHRH analog sort of thing. But the basic mechanism is like initially during the growth phase, FSH will be stimulated, which will go on for production of androgens in males, production of estrogen in females, ultimately leading to spermatogenesis and vitellogenesis. Whereas during the maturation phase, when the fishes are going to ovulate or going to the process of actual spawning, there will be functional importance of LH, which in turn will be stimulating production of maturation-inducing steroid or progestogens, ultimately leading to spermiation. That means release of sperm and oocyte maturation and ovulation or release of ovum. So we can just categorize the reproductive axis into these two phases. During one phase, there is development of the gonadal structures or development of gametes, either through the process of gamete development or gamete growth. And the second step is actually the maturation phase, where the process of release of sperm or the process of maturation of ovum and ovum release happens. And the first step, that is the growth, is being regulated principally by the C18 or C19 steroids, that is androgens or estrogens and androgens respectively. Whereas the next step, the maturation phase, is being regulated by the progestogens, the C21 steroids, that is by these maturation inducing steroids. So if you just look into this functional endocrinal pathway of hormone production, I've just highlighted these two parts, that is the production of 17-beta estradiol and production of 17-20-beta dihydroxy 4-pregnenolone or MIH, that is maturation inducing hormone. So during the initial phase of gamete development, if we consider female, that is more easy to understand, then there would be predominance of 17-beta estradiol. So 17-beta estradiol during the early phase of gamete development will regulate the process of gonadal growth or ovarian growth. Follicular growth will happen under the influence of 17-beta estradiol. Now, once the fish has matured to ovulate and undergo the functional process of maturation, ultimate maturation and release, then there would be a steroidogenic shift from 17-beta estradiol to 17-20-beta dihydroxy 4-pregnenolone, which is the MIH or the maturation inducing hormone. So this steroidogenic shift. So now here comes the integrating part between endocrinology and fishery. So if we want to increase production, so what we can do, we can just manipulate either of these two hormones. We can increase the developmental growth pattern with respect to 17-beta estradiol manipulation, or if we want to induce their functional maturation for ovulation, we can go for production of MIH. So these type of manipulating things can happen or can integrate endocrinology with fishery. If someone has the knowledge of this hormonal background, how the reproductive axis work in fish, they can always go for doing work with respect to increased breeding technique. So we all know about this hypophysation, the induced breeding. So what is that? That's simply inducing the ability of the fish to go for reproduction. So in that case, we are using different type of hormones, the pituitary extracts. The pituitary extracts actually contains the gonadotropins, the LH, which is inducing the differentiation or the functional maturation of the gonads, and the ovum and the sperms are now being released. So the process of spermiation and ovulation is being induced by this particular process of hypophysation. So we all know that there are several these type of different chemicals are being available. Like we all talk about this ovoprime, the ovotide, which are synthetic hormones of fish spawning. There are several brand names of this: the human chorionic gonadotropin, the sumac and the cyanohorin, the ovoprime, the punitazole, the 11-deoxycorticosterone acetate or DOCA. All these are different combinations of hormones. Principally what? Principally of GTH, the LHRHA, the analog of LH, which will increase the secretion of, or which will act like an LH, so that the process of MIH can be stimulated, MIH production can be stimulated, and functional maturation and release of ovum can happen. And they also contain certain dopamine inhibitors or dopamine blockers, as we have talked about. Yes, dopamine is an inhibitory or having an inhibitory effect over the process of gonadal maturation. So if we remove the inhibitory effect of dopamine, then we can stimulate the process of ovulation in fish. So that's why dopamine inhibitors like domperidone are being associated with. So in two ways we are doing this type of things. On the one way we are stimulating LH, on the other hand we are inhibiting dopamine. So the negative regulator is inhibited and the positive regulator is stimulated. Ultimately the effect would be increased breeding product or increased breeding in fishes. So that's is the scope of integrating fisheries with endocrinology. If we don't have the knowledge of this reproductive axis being associated, the endocrinal axis of reproduction, how we are going to increase breeding? So now you can always say, okay, fine, there are all these techniques are being known, but not all is known. So we can look for different other chemicals or different other natural products which can actually act as an alternative for this type of synthetic hormones. Often the use of synthetic hormones are being regarded, okay, not beneficial for the consumers, not beneficial for ecology, for environment. So we are always looking for some environmental friendly natural alternatives to synthetic hormones. Okay, so in these cases, the research is still going on. How to replace, for that matter, this domperidone, or for that matter, LH analog? Can we get some natural product which can act as LH analog? Can we manipulate some other endocrinal orientation by which we can stimulate the process of breeding? And not all the fishes are being associated with this induced breeding. Mostly induced breeding are being done with the Indian major carps, but there are so many other fishes, so many other ornamental as well as food fishes. If we can perform a sustainable process of induced breeding for those fishes as well, so ultimately the total production can increase. So in those cases, melatonin is one important target candidate, I would say, which is a natural product which is often being used in this sort of experiment. Can you use melatonin to increase the process of reproduction or the process of gonadal maturation in fish? Is a very much important aspect of this type of researches. As we can see, by application of melatonin, what we can do is like we can give melatonin which can bind with the corresponding receptors and can cause activation of maturation promoting factor. So melatonin can actually increase or can stimulate the functional activity of maturation inducing hormone. Melatonin, as you can see here, is positively regulating the production of MIH. MIH can then bind with the corresponding receptors and convert the inactive maturation promoting factor into active maturation promoting factor, ultimately leading to the final part of ovulation in fish, that is germinal vesicle breakdown or GVBD. So that's the most important aspect of fish breeding, that occurrence of GVBD or germinal vesicle breakdown by activation of MPF. So that can be stimulated by melatonin. So many different avenues of research can actually be there. We can always work on all these different things. As you can see, this light-dark cycle often being associated with secretion of different hormones. The acetylcholine, the norepinephrine, the dopamine, which is being mostly secreted during dark, whereas norepinephrine and dopamine during light phase are negatively oriented, whereas acetylcholine is being secreted more during light phase. So with respect to all these functional endocrinological portion, we have to balance our fishery part by when to inject. We have all known that during the induced breeding, when to inject the fishes with respect to the hormone. It's not like that whenever we thought of, we just put the injections. It has to be done during the wee hours of the mornings at 1 or 2 a.m. During the night, it has to be done. So whenever these things are happening, that has to be associated with the functional pulsatile secretory orientation of the endogenous hormone. So that's why it is important to have or integrate the knowledge of endocrinology with fisheries. Coming to growth, we all know that, okay, growth hormone is very important. So this gromone is one of the synthetic hormone which is often being used for stimulation of growth here. So from hypothalamus, we know that GHRH is being released, which is positively regulating the pituitary to secrete growth hormone, which in turn is regulating the liver or other tissues to release different type of growth factors, the IGFs, which is associated with body growth, uptake of feed, utilization of feed substances. On the other hand, somatostatin is negatively regulating the process. Exogenous growth hormones can stimulate growth by manipulating all these downward pathway. The point of research would be, often we are a bit skeptic about consuming something which is being treated with exogenous hormone. Okay, this fish is being given injection of a growth hormone and we have increased the size with growth hormone injection. Should we consume this type of fish? Look for some natural alternative. That is one part of research, which try to find out some growth hormone analog which is natural, but and that's why they can stimulate the growth in fish, but because they are natural, they are more biodegradable, so the residual effect won't be there. So such type of research can definitely be done. The pathway of how this type of natural products can actually stimulate growth in fish would be definitely the concept to ponder in case of future research. The process of stress. We all know that stress is definitely a neural stimulation, which is going to affect the hypothalamus through the pituitary and the internal tissue, as well as through the chromaffin tissues. They are affecting the cortisol secretion and the catecholamine secretion. So through the primary response will actually come in terms of secretion of increased cortisol. We all know that with increased stress, cortisol secretion is being stimulated from the internal tissue, and catecholamine secretion from the chromaffin tissue will also be increased. At the secondary level of response, there would be decreased immunological responses. With increased stress, cortisol is believed to be, or it's associated with immunosuppression. They're going to reduce the immunity of the subject. So there would be several metabolic changes, circulatory, respiratory associated systems, which ultimately is going to affect the growth, the disease resistance, and the swimming capacity of the fishes, which are the tertiary response of the stress. So altogether, stress can affect the physiological status as well as growth pattern of the fishes. So definitely there is point to research how we can manipulate what sort of chemicals or what sort of natural products can be given to reduce the stress, to increase, to enhance immunity in fishes so that they are able to withstand stress. We are culturing fish in different sort of environment. We often know that these Thapa culture fisheries, where the wastewater fisheries are very much important for production of fishes. So whenever we are culturing them in wastewater, they are subject to different type of stresses. Nowadays, with different changing environment, we are often find that they are subject to salinity stress, thermal stress, and how they are coping with such type of stress, whether they will be able to maintain their growth pattern, maintain their disease resistant pattern under the circumstances of stress is definitely an important issue of research. So in these cases, we are integrating fisheries with that of endocrinology, how the endocrinal system is related with that much of production. So that's again the same sort of thing, the different type of chemical stressors like the polluting agents, the low level of oxygen, physical stress like handling stress, the capture stress, the perceived stress like startle response, sound, presence of predator. All these different type of stresses are the primary response is associated with increase in hormone levels, which can in turn affect the or cause metabolic changes by increasing in glucose or lactate level, change in immune function, ultimately compromising their immunity. So can we give them some sort of functional food which enhances their immunity? Can we add something to their food, which to their feed during culture that would enhance their ability to cope up with stress, enhance their ability to antigenic pathogenic infestation, and ultimately affect their survival and growth? So those are the important issues associated with endocrinology as well as in fisheries. So during migration, smolting in salmon. So whoever has consumed salmon, they know that it's a very important phenomenon that happens in salmon. The taste of salmon actually comes from the process of this molting, which is very much involved or associated with the change in hormonal concentration. Prolactin is a very important hormone that is acting on gill, skin, kidney, urinary bladder, and making the fish capable of undergoing the process of migration. If Hilsa don't have the mechanism of changing or adapting to the changing salinity whenever it's coming from sea to the estuarine system and from estuarine to the freshwater system of Ganga. So what would happen if they don't have the ability to adapt to the changing salinity? So these changing salinity pattern, we all know that nowadays there is a great project associated with culturing Hilsa in freshwater ponds, which is a bit successful. But the main problem with that particular project is related to this endocrinal aspect, that they need a saline shock to undergo the process of this breeding and to undergo the process of the taste of Hilsa would actually come this salinity shock. So how they are coping up, how this the physiological balance is being maintained through endocrinal system is again definitely a point of very much research, because if we just can find out, okay, this particular hormone is actually orienting themselves to undergo the physiological changes, enabling them to be successful or successfully living or adapting in the freshwater system while coming from the saline system of the sea and again going back to the sea. So then we can manipulate that endocrinal aspect in the freshwater itself and can generate the taste and the breeding ability within the freshwater system itself. So those are the different part of endocrinal research which can be integrated with that of fisheries. So here we can have the different type of research topics which are integrating the fish as a research model or integrating the endocrinology with that of fisheries. Like we can take fish as a research model, like the zebrafish. We all know this particular fish is very much a fundamental model for any sort of studies. Like in developmental biology, you want to do genetics, you can take zebrafish as a model. You want to do toxicological studies, you can take zebrafish as a model. Neurobiology, in case of regenerative regenerative biology, in case of cancer research, in different fields of biology, zebrafish can always be taken as a very good model system. So there are different issues related to why zebrafish is a very good model. You all know that the genome sequence of it is being done, so we can easily manipulate this particular fish. Again, they are transparent in nature, so of course it may happen that if you inject with some say I would say rather substances or dyes, we can just trace the whole pathway of the dye through the developmental process and through the different organ system as well. So altogether, zebrafish is a very good research model. Then, and we can, it's not that everything is being done with zebrafish. You can just think of a new research topic, and you can then extrapolate, or rather think of zebrafish as a model for your basic research and extrapolate whatever you have got in zebrafish to that in mammalian system because of certain similarities as well. Then we can work on breeding manipulation. So whenever we are looking for breeding manipulations, we can just, I can talk about my lab, what we do in our lab actually. We are going to produce a different type of monosex fishes. We can produce, because in different fishes it happens that one particular sex either grows large or they are actually showing better coloration. So even if we consider about food fish, we'll definitely try to culture those who is growing large and growing in a uniform way. And if we consider about ornamental fishes, we would definitely like to culture those which have better coloration. So if we just manipulate the endocrinal orientation in such a way that the fishes are produced in one particular sex in an environmentally sustainable mechanism, then it becomes very much associated with the process of or the development of technique related to fishery, but using the knowledge of endocrinology. So these things, we are using different type of plants here, as we are talking about the natural products and all these things, so that we can manipulate the breeding procedures and ultimately produce some sort of like this type of more colored fishes. You can just look into the male guppies are more colorful, are more marketable compared to the female guppies. The male dove gummies definitely have more price, catch more price in market rather than the females. The tilapia, the males, they grow bigger, so they are catching, or they grow more uniformly, so they are having a better price compared to the females. So all these breeding manipulations can actually be done with the knowledge of endocrinology, ultimately affecting the production in fisheries. We can enhance growth. We can use these different plants which have got some immunostimulating properties, some aphrodisiac properties, thereby producing either one of the sex and increasing the immunity, decreasing or increasing the capability to tolerate stress in fishes. So these fishes, these particular plant materials, they can actually enhance immunological features in fishes. They can make the fishes better adapted to stress. So all these things are very much important part of research. As we're talking about these adaptive physiological works in terms of reproductive biology, environmental toxicology, pharmacology, and therapeutics. So the fishes being cultured under the wastewater system. In the wastewater, they are susceptible, they are subject to different type of heavy metal stress, which can ultimately affect their muscle quality as well as their reproductive efficiency. So can we have some mechanism by which we can say, okay, so after the fishes, if they are being fed with certain type of functional feed, some certain type of nutraceuticals, they would be able to cope with such type of stress in a better way. The muscle quality of those fishes would be better. The growth pattern, the growth ability of those fishes would be better compared to the fish which are not fed with such type of nutraceuticals or such type of functional feed. So these are again being related to the endocrinal aspect. The basic knowledge of endocrinology can ultimately be implemented in terms of fishery. So these different types of physicochemical parameters, the different abiotic stress, salinity, pH, temperature. So we are working on this Arunachal Pradesh project with respect to a particular loach, the Botia rostrata, and we are trying to increase its adaptive orientations. Now that fish is basically a cold stream fish. We are transporting them to Calcutta, or we are transporting them to different places which is not that much cold. So can we give some, or can we manipulate some endocrinal adaptability in such a way that they are able to withstand better the temperature stress? So those sort of differential functional orientation can be a topic of research. How we can increase productivity, how we can increase adaptability in fishes, and thereby increasing the marketability of the fish with the basic knowledge of endocrinology, ultimately leading to the functional orientation in fishery. Okay, now what about the future? Can we go for entrepreneurship? Can it be an option for zoology students, whether they can actually go, whether they can look for fisheries as a career option? Now, okay, that's a negative point, I would say, because we have a separate course in BFSC, the Bachelor in Fisheries, and the MFSCs. And previously, most of us during our time, during 2000 and during the 90s, I would say, many MSc students, they took fisheries and aquaculture as special paper because after having completing MSc with fishery as special paper, they could get jobs as fishery extension officer in different government sector. So in the fishery sector of the government, there is one post, namely fishery extension officer or fishery officers. So these posts were very much coveted posts, definitely. We can get the jobs after completing our MSc with fishery as our special paper. But nowadays, those jobs are being restricted simply because ICAR is providing the course in BFSC and MFSCs, and they are now consuming people who are coming out of these courses, doing BFSCs or doing MFSCs. They have a better chance of procuring jobs as fishery extension officer in different government sector or fishery sector of the government. So, okay, that's definitely, after doing zoology, BSc or MSc, that option is very limited to get directly a job. But you can always do a bit of rather diploma in different topics. Several such colleges, diploma colleges, are also being there. And if somebody thinks, okay, I would like to do BFSC or something like that, they can always go outside West Bengal mostly. Here in Calcutta, there is only one college which offers BSc in Industrial Fish and Fisheries, that is Ashutosh College, and of course that BKC college is also there. Okay, these two are actually offering the BFSC course. But anyway, if you are a zoology student, you can think of entrepreneurship in fisheries. Not necessarily a government job, but you can develop your own pathway for money making, I would say. Rather, as we know that aquaculture is definitely important. There is by 2050, the consumption of animal protein is going to increase by 52%, and of course, fish is going to be one of the very important aspect of such animal protein source. Many of the people are going to depend on fisheries or fishes with respect to the source of animal protein, because often they are cheap but good animal protein source. Okay? So as you can see, the progress level, so that's much more high with respect to fisheries compared to piggery, poultry, or dairy for that matter. And as you can see here, the requirement is also going to very much increase. So the total aquaculture is contributing actually half of the food fish globally. The capture fisheries are past, and aquaculture is actually the future, because we have to depend on culture, because whenever we are talking about capture fishery, we are talking about depleting our natural resources. We are talking about the sustainment or sustainable fishery is definitely oriented with respect to culture as well, not only capture. And we cannot grow with respect to capture fishery, but culture can definitely grow with much more intensification. But the concept of sustainability must be there. So whenever you are talking about sustainability, that means if you do culture in such a way that would also maintain the environmental condition as well. And under the context of this COVID pandemic and all this, where a large-scale operation, a mass-scale involvement of people in aquaculture functioning is becoming very much limited, we have to think of a domestic, local scale of production, individual scale of production. And in this context, the concept of entrepreneurship is becoming more important with respect to aquaculture system. So the ideology is being transferred from this mass scale production involvement into a small scale local production system, and the new techniques being related to aquaculture development. Okay, we're talking about this capture fishery, which is depleting the natural fish population in a very alarming way. So there must be research work conducted so that we can keep a balance between how much to capture, to maintain a sustainability in terms of the capture fisheries as well, to maintain a definite orientation in terms of how much should we capture, when should we capture, what type of fish, what size of fish. All these research topics are to be discussed with respect to this fishery aspect of capture fisheries. There are several ways of fish culture that is being present in your UG syllabus as well. We can actually culture one particular fish through monoculture. We can look for a polyculture system, combining different fishes with each other. And definitely we have to combine these things not only on the basis of, okay, we should go for a polyculture of the IMCs. We all know that from the very basic books, okay, there is a definite ratio of this percentage should be Leviorohita, this percentage should be Katla Katla, and this should be Sinus Mikala. But that's not all. There are many things beyond these three IMCs with respect to polyculture. We can look for different new species, introducing new fish species in the culture system. And researches must be conducted to find out what should be the perfect combination, what should be the ideal stocking density for all these things. Then one very important issue is paddy cum fish culture, the integrated fishery with respect to rice culture, combining them with fish culture. So what sort of fishes can be done to be cultured? Like in Sundarban, so what we are doing in Sundarban is like we are just converting the agricultural fields, because agriculture there is very much dependent on weather, on rainfall, which is the pattern of rainfall is changing. The land pattern is also changing. So you are converting the agricultural fields into aquaculture ponds, mostly culturing shrimps. So can we think of an alternative after all this Amphan and all the shrimp culture system, the production system has gone completely destroyed. Can we think of a system where we can combine this agriculture with aquaculture? We can look for different species which can be sustainably cultured with paddy, thereby increasing the production level. We can think of different other techniques which is intensive, but in terms of manpower, or it's limited. We don't need that much of a huge manpower for this. The setup cost would be definitely high, but once that has been set up, we can use this like this raceway culture, this type of cage culture system. All these new development can be there. The raft culture. And for entrepreneurship, there's a development of biofloc culture system, where the waste from food, fishes, and the urine, they are increasing the ammonia level within a tank. But these ammonia level, by using certain type of biofloc containing different types of nitrifying bacteria, we can just convert them into nitrate and nitrites, which can actually be used up by the fish for production of nutrient substances. So these sort of biofloc system, which is basically biofloc system means where there would be aeration to have a carbon and nitrogen ratio in between 20:1. So that's the main orientation of biofloc culture. So by this system, we are increasing the nitrifying bacterial colony, which will be maintaining the water quality. So you know that with intensive culture system, the problem would be with respect to increased ammonia level in the culture tank. So if we just convert the ammonia into nitrate and nitrites with the nitrifying bacteria, and together with the phytoplankton and zooplankton, which would be consuming those nitrate and nitrates, and there would be a sort of algal bloom, and those algae can act as a additional food for the fishes being present within the tank. So in that way, a typical integrating system can be there. Self-sustaining system can be developed with respect to biofloc. So this biofloc culture of different fishes are now coming into picture, and several training systems are being offered by this CIFE, CIFA. They are offering different types of these training programs with respect to this biofloc culture. So any zoology student, with interest in fisheries can actually contact CIFA or CIFA, these sort of institutes, and look in their websites for such type of training system. And once they are having this training system, they can definitely develop their own entrepreneurship with respect to these things. We can go for aquaponics, which is basically a combination of hydroponics with aquaculture. We can have simultaneously fish culture and along with production of certain plants. And the beauty of this particular system is like they're again self-sustaining system. We are using and we are recycling water. So in terms of whenever we are talking about reduction in the freshwater level, the third world war is going to be happening with overwater, or the sources of fresh water, and the freshwater sources are being limited day by day. So these sort of systems where resource utilization is being very judicial in nature. So there is recirculating mechanism where the same water is being purified through the hydroponic system and again being recycled for aquaculture is very much important here. Again, aquaponic systems, these different institutes like CIFA, they are offering such type of training programs. We can always look for those and can develop our own entrepreneurship. This particular figure is definitely not my lab, but some other lab where they're within the lab, they are set up on this particular very beautiful aquaponics culture of plants along with certain ornamental fishes. So not necessarily all this has to be done with large scale food fishes. So you can go for small scale ornamental fish culture as well with respect to type of aquaponics or biofloc culture system. Research with respect to transgenic fish, of course, it's an advanced area of fishery research. So many different type of transgenics we all talk about. The glowing fishes, increasing the fluorescent development of fluorescent fish. They look better definitely in the aquarium, fluorescing in the night. And the developmental, biological, and endocrinal studies being associated with the process of development of transgenic. We can increase the production level with respect to transgenic as well. Several transgenic tilapia are being developed Other carbs are also being developed with respect to transgenic, increasing production. I won't be going into the ethical issues of producing transgenics, or that is a separate debatable thing. But anyway, transgenics are in, so they are being produced, and that is also a definite concept of research in combining endocrinology genetics with that of fisheries sector as well. So we can always look for this fish biodiversity and fish economic system. So these things are again very much advanced stages of fishery development or fishery research, where people can just go out, not necessarily looking within India, but abroad as well. So we can look for monitoring the fish diversity, the pattern of migration, which effectively can be channelized for increasing fish catchment by the different sea traders as well. So combining all these things, the farming, technology, the medicine management system, so we can go for research after doing our BSc or MSc, again doing the same thing with NET, GET or theory. And I don't think like in foreign countries there is no scope of research in fisheries. Definitely there are. Israel, the Scandinavian countries, the Norways and all this, they are very much advanced in fisheries technology, and they do research mostly in marine fisheries, definitely. But Israel is one of the countries which is doing a very good thing with respect to freshwater aquaculture as well, in association with marine fisheries. Then, of course, the Southeast Asian countries are also doing very good sort of research work, like the Vietnam, Thailand. They are also doing a very good research work in fisheries as well. So we can look for scopes not only in America, but also in these countries. I'm not mentioning China, but of course, China is also doing a very good research work in fishery sector. They are actually the highest producer, both in terms of freshwater aquaculture as well as in terms of marine aquaculture system. China is by far the highest producer of fishes in the world. So several research works are going on in those countries as well. So if you have the zeal of doing research work in fishery sector, you can always look for the websites of these different institutes, whether they are offering any positions, be it at the PhD level, be it at the postdoctoral level as well. So here are some of the universities in marine, or mostly marine, as we have talked about: the Swiss Federal Institute of Technology, MITs, Harvard, Cambridge, Oxford. So, okay, these are the best ones, I would say. But don't think about, okay, I have to get admitted in these particular universities only. But all the other countries I have mentioned, they are working very good in terms of fishery research. So you can always look for some upbeat countries as well to look for research options with respect to fisheries research. And if you want to stay here and develop your own entrepreneurship with respect to fishery, with your background knowledge of zoology and with your background knowledge of endocrinology, you can always give input to these different type of NGOs and small scale groups with your knowledge, so that they can, with your knowledge, the marginal, the terminal people, they can also be helped with your effective knowledge as well. So such type of entrepreneurship, such type of limited local scale involvement of people and your orientation can actually help development of not only you, but as a whole, the society as a whole. I'm finally at the end of my talk, so I'd be just okay. If you do want that, or you're telling all these things, but what's there in doing research work in fisheries and endocrinology? We always say, okay, genetics, okay, we are going to do PCRs, we are going to run jails and all these things. Those are much more coveted things, doing works in the labs and finding, sequencing, all these things. Can we do this in fisheries or do this in endocrinology? Okay, not necessarily endocrinology or fishery is not entirely genetics, but you can always combine these things. If you want to do fish genetics, you can take fish as a model and do all these techniques in whatever lab you are working. So as I have told, the zebrafish is a very good model for doing any sort of work. So you can use all the latest advanced technologies, taking zebrafish as a model. So ultimately, you can, if people ask what are you doing, you have to say I am working on fish. But then you can add, okay, no, but I'm working on fish, but fish as a model. I'm doing it a whole lot of advanced technologies. Okay, jokes apart, we do in our lab different types of bacterial culture, different immunoassays for that matter. The ELISA, more precise, the immunoblottings, the Western blot principally. We are thinking about doing PCRs and RT-PCRs. We are again doing some sort of chromatographic separations, TLCs, column chromatography. We are doing this atomic absorptions spectroscopy as well. Basically preparing the samples here and doing the AS in some other labs which have the machine. Basic histological works are being done, and the spectrophotometric studies, the immunological studies are definitely being done. And the park is, we also do some field work. So if anybody is interested in traveling, in moving around in fields and all this, okay, fishery would give you that scope as well. So if you can integrate endocrinological research with fisheries, you can have a scope of working in the lab as well as in the field. So someone who is interested in both lab work and in field work, he can think of choosing this particular topic of fish endocrinology or research in fisheries integrating endocrinology with it. It would get he would be getting a scope of doing the field work or traveling in the field as well as in the working in the lab as well. So these are all different fields that I have, but I'm not advocating that you should come to my lab, definitely not in my lab. But anyway, you can integrate fisheries and endocrinology in such a way that both your orientation for lab work and your zeal for moving outward or to look for field work, to go beyond the lab, will be satisfied, will be fulfilled with such type of research. So this is my team who has helped me a lot in preparing all the slides, giving me all the data, giving me all the information. So it's a big thanks to them, definitely, and thank you. So that's saying. Now we are open for any question.
Thank you very much, Shuman, for such a nice explanation and for a pretty long lecture, which is really very enthusiastic, and it also contains a lot of information regarding their career. You have integrated really both the career as well as the advanced learning in the field of endocrinology and fishery. Now, I would like to ask you to off your sharing screen and come to the screen. I mean, we want you visible on the screen. You please on your
Okay,
video.
fine. Let me just
And participant,
I will stop sharing and
yeah.
Just
Participant, this is an announcement. I have already posted the feedback link over here, and we will continue this meeting so that you can collect the feedback link for 15 minutes after the completion of this discussion session. Shuman, please on your video kindly. Now, you participants, if you...
I'm just trying to start my video.
Okay. If you have any...
I've stopped sharing.
Yeah, you have already stopped sharing probably. Yes, you are on, but I cannot see you.
Yeah. Yes, I've stopped.
Yes. Okay. Now, participants, if you have any question or anything you want to share with sir, you can unmute yourself and make your question clear, because your feedback form also contain assignment. Okay, your feedback form also contain assignment that you have to do and that you have to provide or submit to us. That Google form is to be submitted to us by 12 today night. And each and every day this kind of assignment is very much necessary, because your certificate will be engrossed not only by Vijay Krishna Girls College Cytogenetics and Genomics Lab, but also by Clarkson University. And after each session, within 24 hours, we have to send some attendance proof over there, on which they will actually give us the certificates. Okay, so that is very much necessary, not only for certificates. Those who are interested, these are very, very small assignment that you have to do. So I have some more declaration, but before that, if you have any question, feel free to ask sir. Sir is there. You can unmute yourself. Whether you can unmute, you can unmute yourself.
hello, sir. So, am I audible?
Yeah.
Yes, sir.
Yes,
Sir,
you
I
are.
want to ask one
So what is your
question.
question?
Yes, sir. Sir, as you have shared one of the slides showing that there is a threat of population degradation of ornamental fishes. No? So, sir, the reason might be the increase in population of the humans, like increase in demand of the fish food. So, I want to ask that is there any threat in the population degradation of ornamental fishes also?
Uh, Please, can you repeat your final questions? Is there?
Sir, I want to ask that is there any threat in the population degradation of ornamental fishes also?
Okay. In case of ornamental fishes, okay, is there any feed which can enhance coloration? That's what you're asking, isn't it?
Yes, sir. Yes, sir. Yes,
Yeah,
exactly.
yeah, of course. We know that the pigmentation pattern is often dependent on carotenoids. Okay,
Yes.
so if we can add different types of carotenoids, okay, and carotenoids can be obtained from different plant product as well. So different plant leaves or the barks of different plants or different algae, they often contain different quantities of carotenoids. So we can look for developing certain functional food uh, by adding those plant products or by adding carotenoid itself to the fish feed and being the fish, the ornamentals being fed with such type of feed can enhance the coloration pattern. This is again a very much a well topic of research as well. What type of food to give, because not necessarily all the ornamental fishes will consume same type of food. Okay? So often we are finding that the products, the fish feed which is being sold in the market, they give that. If you look on those packets, you'll be seeing that it's they contain carotenoids. This much percentage of carotenoid is being present. So you can think of manipulating these percentages of carotenoids and ultimately think of increasing the coloration pattern. That is also a part of research.
Okay, sir. So one more question, sir. I want to ask your views, sir. So which would be the better option to go with the fish food culture or go with the ornamental fish? If a person want to start his or her personal entrepreneurship, so which one would be the better option, sir?
Okay, fine. So that depends on what you actually have. So if you consider culturing fish in your domestic system, uh, I would say rather it's better to go for ornamentals because the in domestic system there is definitely limitations of space. Okay, so whenever you are trying to build a tank or rather you are trying to build a system with respect to this
Sorry, sir, I'm not getting you, sir. It might be due to the
bacteria.
network problem.
So it's better to have a ornamental fish culture rather than food fish culture. Food fish culture is
Hello.
you just cannot produce one or two. It has to be a large number of fishes if you want to start an entrepreneurship with respect to food fish. But what you can do is
Shuman, may I request for one thing? Interrupting, extremely sorry, but
then food fish development of food fish.
Dr. Chakraborty, I am
Huh?
just, Shuman, I am interrupting you. Probably your net speed is not okay. So you put your video off, then your audio will be completely okay for the participant. Otherwise, it is cracking.
Okay, fine. So
Uh-huh.
cracking. Okay, so I'm stopping the video.
Stop the video, then the bandwidth will be okay for interaction. Yeah, now just continue. We will see.
Okay. Okay. So in those cases, I would say development of entrepreneurship with respect to fish feed development is fine in your domestic system because that would again comparatively you can genuine opinion from my regard. I would definitely say okay, it's better with ornamental fisheries for development of initial small scale entrepreneurship rather than fish feed culture. But if you have say one or two ponds to spare with, definitely you can look for food fish culture.
Okay, sir. Thank you, sir.
Is there any question from the participants? Please ask to Dr. Chakraborty. Participants, you may ask freely to Dr. Chakraborty. Participants, do you have any other question?
Hello.
Hello.
Sir, am I audible, sir?
Yeah, you are audible. Please introduce yourself first. From which college you have participated?
Okay, sir. Sir, I am Shomrup Chakraborty from Department of Zoology, University of Calcutta, second semester.
Okay, Shomrup.
Sir, is there any marketized endocrine tool that we have to reduce stress, handling stress or transport stress on fishes that are sold in markets in India?
The first part of your question, I get that you are talking about the handling stress which are being of other fish being sold in markets of India. But what is your actual question?
Sir, do we have any endocrine tool or endocrine medications that are marketized for those who actually sell the fishes, so
Okay,
that they
for
can
reducing stress in
yeah.
okay, so certain type of drugs or something which can be used to reduce stress. Okay, there are often different mechanisms by which you can use or which can be used for reducing stress. One very common thing, as you have seen, whenever the fish are being marketed. Have you seen the trucks or the matadors carrying those fishes in a hurry and all these things, and always putting their foot inside the bucket or putting their hand inside the bucket and generating a sort of aeration. So that is one form of reducing stress by creating aeration within the system. But of course, if it's a very large scale system, there are definitely tanks associated with say aerators for that matter. But the fishes are often being given sedatives. Certain type of sedative drugs are being used so that the fishes they are being sedated while transporting, and they don't go for any sort of that much of a movement and everything, reducing their stress to a certain extent. This is pretty common aspect as we are doing in for transportation of other livestock as well. Mostly, so if somebody have transported their pets from one place to the other via air cargo, they would know that the pets are being given sedatives so that they make that is less stressful for them as well. So that same principle is being applied for fish as well. Often different types of sedative drugs are being used. That can be chemicals. Sometimes that can be long oil, lobong tel যেগুলো হয়, the cardamom oil. Those are being given as sedative substances. Temperature may be reduced to a certain extent. We can add ice to the water. The reduced temperature can give a bit of a sedative effect over the fish. So different techniques are definitely there to reduce the amount of stress. But what I'm talking about here is if you just can increase the adaptive ability of the fish to withstand such stress, maybe it's better for them as well.
Okay, sir. Thank you, sir.
Okay, Shuman, I will be happy to receive more questions from the undergraduate colleges, because post-graduation students, particularly from the University of Calcutta, they have opportunity to interact personally with the Dr. Shuman Chakraborty as their teacher. So if there are some questions from the undergraduate colleges, I will be happy to receive. Is there any question, student from the undergraduate colleges? Please come forward to ask questions. It's an opportunity to interact with the such a doyen personality in the field of fishery from University of Calcutta. You please ask question freely. Don't hesitate to ask. Even you can speak in Bengali also if you have problem in the speaking English. So don't hesitate, come forward. Students from the Bijay Krishna Girls College, this is your program, your institution actually hosting this program. So it's your responsibility to ask any question to the resource person. So students from other colleges as well as Bijay Krishna Girls College, please ask freely any question that come in your mind to Dr. Shuman Bhushan Chakraborty.
There will be a special certificate for the students who will really interact highest throughout all the sessions. I must declare there will be a special certificate for the student who will interact rationally highest. There are some questions in the chat box. I would like to ask Amrita or MM to take questions from the chat box and tell those questions to Shuman. May I ask Amrita or MM to on their video and take questions from chat box? May I ask her? There are a lot of questions in the chat box also, I can see. MM, can you continue those questions?
Okay, I'm fine.
Shuman, 1 or 2 questions we are taking from chat boxes. Okay, then we will complete the session.
Yeah, definitely.
Is that okay? Is that okay? Because you have already given a very informative, large lecture. So will it be okay with your kind permission?
Yeah, of course.
Okay.
I have no issues in answering questions from the students.
Ma'am, am I audible?
Who is that? Amrita?
Amrita, हाँ.
Yeah, you are absolutely audible.
हाँ. One question from Tuhina Jana. We use domepridone to get rid of nausea. Then why we use in fish and what is the working mechanism?
Okay, domperidone is actually a dopamine inhibitor. Okay, of course, dopamine inhibitor has different type of functional activity depending on the different type of functions of dopamine itself. So whenever a particular person is suffering from nausea, so we use dopamine inhibitor to reduce the concentration of dopamine in the system. Dopamine, rather, it's a thing which keeps us awake. Okay, so in those cases, dopamine inhibitor is actually affecting that physiological functional activity. But whenever we are using domperidone as a dopamine inhibitor in case of fishes, we are inducing reproductive orientation. As I have told you that dopamine is an inhibitor for LH secretion. Okay. So what dopamine does, the signaling from dopamine, it prevents secretion of LH from pituitary. So in those cases, reducing dopamine secretion will actually increase the propensity of LH secretion from pituitary, which in turn can go for giving a positive impetus for reproduction in fish. That's why in association with LH-RH analog, which is altogether stimulating function of LH, we use domperidone, which is a dopamine inhibitor. ব্যাপারটা দুদিক থেকে আমরা ভাবতে পারি। একদিক থেকে আমি যেটা ইনহিবিটর ছিল ডোপামিন, তার এটাকে কমিয়ে দিচ্ছি, আর যেটা পজিটিভ এফেক্ট দিচ্ছিল LH-RH, তার এফেক্টটাকে বাড়িয়ে দিচ্ছি। So that's why we are using domperidone here. The use of domperidone for our purpose is completely a different physiological activity here. Okay? Is it clear or?
যদি কারোর মনে হয় বাংলায় বলতে সুবিধা, কোনো সমস্যা নেই কিন্তু, বা কোনো জায়গা বুঝতে অসুবিধা হয়েছে সেটা বাংলায় বলতে পারো। Okay, if no more question, then
I have a question. Shuman, I have a question. Right. I came to know that Hilsha is being cultured in some area of West Bengal. I don't have an idea whether that cultured Hilsha will have that kind of taste and size and quality of flesh that are caught in from riverine system.
Okay, that's a very good issue, rather than controversial issue as well. So what I have got from Oshinda is actually the Shivgan University. We were talking a few days back in this regard, and he was saying that yes, they were the group of Shuman Bhattacharya sir and all this. They were working on producing Hilsa in freshwater, and they're actually what he has said, what Oshinda and his group has said. Yes. In Noihati, some farms are there who are able to produce Hilsa in freshwater. But the problem is in terms of growth and in terms of taste. So that saline shock is very much important for development of taste in the muscle of fish. Whatever taste we get, that's because of এটা মনে হয় দেখবে যে একদম সমুদ্রের যে ইলিশ মাছগুলো থাকে সেগুলোর থেকে আমরা কেন এই পদ্মার ইলিশ রূপনারায়ণের ইলিশ এইরকম কথা বলি আর কি, because once they travel so much distance, that's very much important for their physiological alteration or muscle quality টার একটা alteration কিছু ঘটে থাকে, which is still beyond the scope of freshwater culture. দুটো problem ওরা আমাদেরকে বলছিল যে they are now facing because the cost being associated with such culture mechanism, প্রোডাকশন করলাম কিন্তু যদি তার গ্রোথই না হয়, মানে সেই আমি ছোট ছোট বাচ্চা ইলিশ রেখে তো লাভ নেই, the growth manipulation is not being done, there is restricted growth in ponds and the taste is another issue which yet they are not able to overcome. সুতরাং okay, I would say they are also optimistic that okay fine, they'll be able to produce quality hilsa in a few years time or so, but that's sort of research. আমাকে যদি বলো, I'm very I would always say okay, that's not going to happen because the physiological impetus is definitely one of the great issues. This traveling, otherwise তো আর হতোই না।
রাইট রাইট রাইট
তবে মানে নৈহাটিতে তো বললো যে হ্যাঁ ব্রিডিং করেছি আর কি, যে ধরে আগে যে ইস্যুটা ছিল যে তুললেই বাঁচবে না সুতরাং ব্রিডিং করতে পারবো না, they have overcome that.
সেইটা ওভারকাম করা গেছে, মানে সেটা সেটা
হ্যাঁ, কিন্তু ওই টেস্টটা বা গ্রোথটা হচ্ছে না, মানে গ্রোথ না হলে প্রবলেম সেটা হচ্ছে তোমার কস্ট এফেক্টিভ কখনোই হবে না, কস্ট এফেক্টিভ
এক্স্যাক্টলি
একটা সমস্যা।
তো ইটস নাইস,
এইখানে তো হরমোনটা ওকে প্রোলাক্টিন আনতার সব কায়দা যদি করা যায়, সো দ্যাটস ইম্পর্টেন্ট. It's a good field of research.
That's another way of research, another way, another avenue for researching in future. So students who got that opportunity to hear from a fish endocrinologist and fish biologist, and such a nice lecture, Shuman, you have delivered. It's a so long 2 hours and highly energized lecture, and I think it's very appealing to the students, particularly the undergraduate students, to get a total glimpse about the fishery and its link with other subject as well, starting from the economic zoology, from genetics, from endocrinology, from physiology, all about. You have given a total overall holistic approach to understand the fishery nowadays and its application in the present day, particularly in the economy. So I think students are extremely enriched with those informations for last 2 hours. And if there is no questions, may I ask Dr. Papia Ghosh to conclude the session for today.
Okay. So thank you, Dr. Ghosh. So I just declared the conclusion, but before that declaration, I would like to tell one thing, that tomorrow our session will be from 7 p.m. to 9 p.m. as it was declared in our flyer. We have already declared in our flyer that there will be some rescheduling of the lectures according to the availability of the speakers and everything. So tomorrow we are going to send you the new link. The old link will not work. We are going to send you a new link in the webinar mode, where you should join by just 10 minutes ahead. I mean, if it is from 7 p.m. tomorrow, you just join at 6:50 p.m. tomorrow. And tomorrow there will be a class on statistics. There will be few basic classes on statistics, so that our overseas collaborator, that is Dr. Shumana Mondol, can show you the application of some basic free software to calculate those statistical method. That's why we will go through some basic statistics as it is revealed in your program schedule that I have provided. Okay, and mind it, it is specially designed for the undergraduate students, so few classes may be
not so much worthy for the postgraduate student, but still the postgraduate student also have to complete their assignment to get their certificate. Few classes, not all classes, but undergraduate students, you have opportunities, you will have ample opportunities to understand statistics, some of which are included in your syllabus, some of which are not included in your CBCS syllabus, but highly needed if you want to pursue higher studies, at least MSc and everything. Then you will get problems. So that this kind of statistical workshops or classes will really be helpful for you, that will be conducted. So that's all for you. I do congratulate all of you for your kind attention, but always keep yourself in unmute. Tomorrow, obviously, we will keep yourself unmute. You can raise your hand, then we can take your question. I mean, we will mute you, and when you will raise your hand, we will unmute you, and you can interact. No problem. So tomorrow, the session will be from 7 p.m. You have to join by 6:50. Please join the Telegram group. I have delivered, or rather we the organizer have delivered mail to all of the registered candidate. Please join the Telegram link, because all correspondence cannot be done regularly in the mail, because it is a long 2-week program. So naturally you are going to get a heavyweight certificate. That's why you must join to the Telegram group to get the material of the resource person, to get all other instruction. That is an apple from our side. Up to this, good night everybody, and thank you Shuman very much.
Oh, it's my pleasure, Papyadi. Thank you. Thanks a
Thanks
lot.
a lot.
Thanks a
Thanks a
lot.
lot.
Bye bye.
Okay.
Bye bye all.
Bye. Bye bye.
Dr. Ghosh, you have to end the meeting because you are the main host. That particular laptop is the main host. Mine desktop is co-host.